The seas surrounding Sabah
Ladies and gentlemen, the virtual talk is going to start in 5 minutes following the video introduction of both institutions. Please be prepared and turn your microphone off. Dear academics communities of Pajajaran University and Malaysia Sabah University, dear guest speakers, Dr. M. Tamrin bin Muhammad LIL, Dr. Rofi Gradiosa, and all of the participants wherever you are. It is an honor to me to inform you that among us here we have the participants from Indonesia and Malaysia. Assalamualaikum warahmatullahi wabarakatuh. Good morning to you all and welcome to International Aquaculture Talk Series 1. My name is Adinda, the host of today's talk. This talk is a part of collaboration between the Faculty of Fisheries and Marine Sciences, Pajajaran University, Indonesia, and the Borneo Marine Research Institute, University Malaysia Sabah, Malaysia. We are delighted to welcome you to our first series with the theme, Fish Health and Disease Management. Ladies and gentlemen, we are now going to opening speech delivered by the head of Master Program of Fisheries Sciences, Faculty of Fisheries and Marine Sciences, Pajajaran University. Please welcome Dr. Iskandar.
Assalamualaikum warahmatullahi wabarakatuh. I am Iskandar, the head of Master Program of Fishery, Faculty of Fishery and Marine Science, University Pajajaran. I want to thank Dr. Siti Rayana, Dr. Hui Ching Abdullah for your partnership in communicating this important event today. Dr. Muhammad Tamrin and the organizer and attendant from the Borneo Marine Research Institute, University Malaysia Sabah for participating this event. And I want to thank all of you, all participants from many backgrounds. Aquaculture is the fastest growing food production sector globally, with production projected to double within the next 15 until 20 years. As we know, the Indonesia and Malaysia has the same climate and region. The cooperation between these two countries has been developed in many sectors, including in aquaculture. To overcome this, we may conduct some collaboration among institutions and aquaculture, and by this joint talk, the opportunity of collaboration between two universities may happen later, such as joint publication, joint lecture, joint research, student and lecture exchange, and so on. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for the opening speech. Now, we are going to listen to the opening speech delivered by the Director of Borneo Marine Research Institute, University Malaysia Sabah, Dr. Siti Raihana. Please welcome Dr. Siti Raihana.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much, Ms. Adinda. To all participants, our counterpartner from University Pajajaran, Faculty of Fisheries and Marine Study. Congratulations! I want to say congratulations to both institutions, especially to the organizers. I believe it is started by Dr. Fitri and Dr. Fui. It's a very excellent effort by the two of them. And I think this initiative is a good start for both university. And I hope in the future, despite of this difficult time due to the pandemic, we can only be what do you call it, organize this activity. But inshallah soon, when it's over, the pandemic over, we can meet physically and then organize more, activities together. But as a good start from these two institutions, I want to congratulate you again. I want to wish you all the best to the speakers for today, Dr. Muhammad Tamrin and also Dr. Rafi. So I think without further ado, we should start the talk, and again congratulations to all the organizers. Okay, thank you very much.
Thank you for the opening speech. Before we start it, we highly appreciate your attendance, and please don't forget to fill the form of attendance today in the discussion session of today's talk. So please wait for the link. Ladies and gentlemen, the next session is the panel session with two speakers, Dr. M. Tamrin bin Muhammad, LAL from UMS, and Dr. Rafi Gradiosa from UNPAD, and will be moderated by Dr. Fitri M. Pratiwi from UNPAD. Dr. Fitri M. Pratiwi is a lecturer of Fisheries and Marine Science Faculty, Pajajaran University, and she graduated from Mie University, Japan for her PhD. Her expertise are aquaculture, fish nutrition, and fish physiology. Now, without any further ado, we will turn the time over to the moderator. Dr. Fitri M. Pratiwi, screen is yours.
Thank you very much, Ms. Adinda. Assalamualaikum warahmatullahi wabarakatuh. Good morning to you all. Thank you, Dr. Siti, Dr. Fui, and all participants from Malaysia and also from Indonesia. I'm the moderator of today's talk. So this is the collaboration between Faculty of Fisheries and Marine Science, Universitas Pajajaran, Indonesia, and also the Borneo Marine Research Institute, Malaysia. So it will be the good start. So this is the first series of our joint talk with the theme of fish health and disease management. So there is two speakers today. So the expertise in fish health and disease from both universities. The speakers are Dr. Tamrin bin Muhammad LAL. Hello, good morning, Dr. Tamrin. Dr. Tamrin.
Good morning.
Okay, good morning. Okay. And also Dr. Rafi Grandiosa from UNPAD.
Hello.
Hello, good morning.
Morning.
Dr. Rafi, are you ready?
Yep. Let's go.
Okay, just a little housekeeping before we get started. For convenient audio visualization, please turn your microphone off during the presentation of the speakers. And the regulations of our talk is speaker will be present for their materials for about 30 minutes. And also we have another 30 minutes for discussions. If you have any question during the presentation, just put in your chat room box and format it with your name and institutions. I will bring that in during the presentation, and we will discuss it in the discussion session. Okay, now allow me to welcome the first speaker, Dr. Muhammad Tamrin bin Muhammad LAL from UMS. Okay, he is a lecturer and researcher at Borneo Marine Research Institute, UMS, Malaysia. He got a doctoral degree from University of Malaysia Sabah in marine biotechnology and also master degree in aquaculture. His expertise is marine biotechnology and microbiology, fish health and disease, immunology, and pathology. So he is a lecturer also and researcher in same institutions. So without further ado, please, Dr. Tamrin bin Muhammad, screen is yours.
Okay, thank you very much, Dr. Fitri. I hope everyone can hear me now. Okay, Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Wassalatu wassalamu ala asyrafil anbiya wal mursalin. Sayyidina Muhammad wa ala alihi wa sahbihi ajmain. Assalamualaikum and good morning, everyone. Welcome to our talk for this morning today. So, on the theme of fish health and disease management, I would like to congratulate the organizing committee for organizing this series, the talk series. And I feel honored to be invited as one of the speaker for this morning. Okay, so let me share my screen first. Please allow me. Okay. So I hope everyone can see my slides now. Okay, I believe so. Basically, the
Yes, sure.
topics that I want to present today is about the biocontrol approaches in aquaculture. So basically, under the fish health and disease management, this is a site area that is actually a small site area that's under the fish health management. So before I start my presentation, maybe I add up some more information on the backgrounds. So the name I think already pronounced by the moderator, Muhammad Tamrin bin Muhammad Lal. I am the senior lecturer in Borneo Marine Research Institute, and also currently I'm handle the as a postgraduate coordinator for this institute. So my research field is mainly on marine biotechnology, but I have a huge passion in microbiology, so I love micro very much. So my current research is about this. It's a combination between the disease and the control strategies. First is on the fish disease. Start with the white PC syndrome is one of the devastating disease in shrimp industry. And then I'm also doing works on the bacteria, which is specific for the bacteriocin producing bacteria. And then the antifungal activity of the seaweed extract, this one to control the fungal infections in crab larvae. And I also do works on the marine omycetes infections in crab larvae. And lastly, my latest works basically is on postbiotics for bacterial disease treatment. So, okay, so this one is just a brief introduction of myself lah. So we move to the topic itself, biocontrol approaches in aquaculture. I would like to start the topics with the control of diseases, which is the health management in aquaculture is important due to the problems such as disease outbreak. So basically, this disease outbreak is one of the major limiting factor in aquaculture. And this disease outbreak usually caused by viruses, bacteria, fungi, and parasites. So these four organisms is basically that cause the is very infectious for aquaculture. Previously, antimicrobial agents and chemical control strategies is used to control the disease, but there are drawbacks by when you use these things because they're creating environmentally detrimental effects to the environments. Okay, and then they also promote the development of antibiotic resistant bacteria. Okay, so that's why I'm going to introduce the biocontrol. So this one is to explore different approach in terms of prevention and treatment for the bacterial disease, for the diseases in aquaculture. For the biocontrol, usually they are safe and environmentally friendly. We can define the biocontrol as the use of natural or modified organisms or its products to reduce the effect of the pathogen. So basically, it's just an alternative to the current control strategies to substitute the usage of the chemicals, synthetic chemicals, sorry. So basically, we go to how to control the disease, basically. So we go with to control the disease, we need to understand the interaction between the pathogens and also the pathogens and also the culture system. So how they interact, then after we understand how they interact, then we can control them. So For the interaction between the pathogens and our culture system, basically the fish or the marine organism or any organism that we culture in our system. First is the pathogen is outside the culture system. So this is the first interactions, whereby the disease can occur when the pathogens enter your culture system. So in order to prevent them, of course, we need to prevent the entry of the pathogens. I believe everyone here understand and have heard about the biosecurity. So for the biosecurity is a process to to prevent the pathogens from entering the system. So if we understand the way how they enter the system, then we can do the prevention. Usually we will look into how the vectors, because the vectors is the method or the vehicles that bring the pathogens into your culture system. So the vectors can be water, of course, because we can do the water exchange. So the pathogens can enter our culture system through the water, and then the feed. Okay, so sometimes because the fish will feed with the fish feed. Sometimes those who are using the pellet feed, that should be no problems because it's low the risk of the contaminations. But if you use the like food fish feed, fish, fat fish. Okay, usually previously we use the term trash fish, but when you use that one, sometimes these fish, they contain the pathogens and they will allows the transmissions to the culture system. And then the other animals, this one, if your culture system is like open sea in net cages, so basically it's exposed to many things. So basically the other animals can transmit the disease also, or the pathogens. Tools and the workers, of course, if they are a lack of sanitations, disinfections. So basically the tools and the workers can also transmit the disease. There are other things, but I maybe don't have to elaborate it more in here. So this is one of the interactions that causing the disease. Second, the other interaction is the pathogens already resides inside your culture system, meaning that they already live over there. In the hatchery, usually we can control this situation, but if it's in the earthen pond outside or in the cage culture, basically the fish already live with the pathogens because some of the pathogens is a normal flora in the environment. So what we can do is we prevent the pathogens from infecting the fish. That's the only way we do it. How? We maintain the good environment so that the fish is not stressed, because if the fish stressed, they will become more vulnerable to the disease infections. Second, we improve the fish health. Of course, this one, we make the fish stronger through the vaccination, provide them with the immunostimulants, and also we can use the probiotic. And third, basically this one is more advanced. We develop the disease resistant fish. It's either through the crossbreed to make better fish, okay, like the hybrid grouper, because sometimes the hybrid grouper is fast growing and some of them are resistant to certain disease. And then, or either GMO, genetically modified organism, or genetically modified fish. Okay, but I won't talk much about the disease resistant fish because this one involves many other things like genetics. So for the biocontrol aspect about the how to prevent the pathogen inside the culture system, I'm focusing on the improving the fish health. So the biocontrol to improve the host health against the pathogens. First, okay, through the prevention method, vaccinations. So basically, if you remember the definition that I mentioned just now is the biocontrol is the use of the bacteria, the organisms, either live or modified, natural or modified, or the products. So basically, the vaccine usually actually a product from the pathogen itself. So vaccines are various preparations of antigens derived from specific pathogenic organisms that are rendered non-pathogenic. They can be into two types, which is either the inactivated vaccine or live vaccine. Inactivated vaccine is like for example, if the bacteria is already being killed, like formalin killed bacteria, that one is inactivated vaccine. For the live vaccines, usually the bacteria is still alive, but become attenuated or weakened, or the pathogenic properties is already been removed from that particular bacteria. So the vaccine will stimulate the immune system and increase the resistance to the disease from the subsequent infections by the specific pathogens. So let me share on how vaccine works. Basically, this one is just simple schematic diagrams. So basically, the fish, we vaccinate it with the vaccines from the specific pathogens, and then the vaccinated fish will develop the immunity against the pathogens. When they encounter the pathogenic or live pathogens, the fish will produce the antibody that recognize that particular pathogens, and then before the pathogens can multiply or doing their works inside the fish already destroy them. The antibody will disable the pathogens, and the pathogen will be cleared by the immune cells. Sorry. So that one is about the first vaccine. Second, immunostimulant. Okay, immunostimulant are available for the control of fish disease and may be useful in fish culture. Okay, the immunostimulatory effect of glucan, chitin, lactoferrin, and levamisole for fish and shrimp have been previously reported. Okay, and then the nutritional factors such as the vitamin B and vitamin C, growth hormone or prolactin have been also reported to be immunostimulated because this thing able to stimulate the immune response of the fish. These immunostimulants mainly facilitate the functions of phagocytic cells. So basically, this one is referring to the innate immune systems of the target organisms, the fish, and then increase their bactericidal activities. And then several immunostimulants also stimulate the natural killer cells, complement lysozymes and antibody responses of fish. So how they works? Well, the fish usually we provide the immunostimulant through the feed. So basically the fish ingests and digests the immunostimulant. So when the fish, when they given the immunostimulant, the immune cell activity is increased. When they encounter the pathogens, okay, the immune system will be activated. So this one I put P and L is a representative for the lysozyme and also phagocytic cell basically. So sorry. So before the pathogens can do their works, multiply, cause the harmful infections to the fish, the immune system play the pathogen first because it's already been activated. So basically, this is how it works. Next, probiotic. Okay, so this one what I like the most because this one is related to the microorganisms. Probiotics are usually live microorganisms which, when administered in adequate amounts, confer health benefits on hosts. Nowadays, probiotics are also become an integral part of the aquaculture practice. So to obtain high productions, of course. So the common probiotics like the lactobacillus, lactococcus, and etc. is become the one probiotic candidates that is used in the aquaculture. So basically here I share with you one functions of the probiotics. Usually for the probiotics, it can be added into water or you can mix with the feed. So this one, the examples is mixed with the feed. So the fish ingests the probiotic. So the probiotic will be reside inside the gut of the fish. So the fish supplemented with probiotic when they encounter the pathogens. The pathogens, when they enter the fish, they will multiply. But due to the the probiotics already colonize the gut, the probiotic, they outgrow the pathogens. So the pathogens cannot multiply. So in that case, it will minimize the numbers of the pathogens, and then the pathogen won't able to cause disease to the fish. Okay, so there are other functions of the probiotics, but I think I don't have to discuss it more here. Later, if we have time, we can discuss more. So, and the other prevention method is symbiotic. I believe some of you may have heard this or may not have heard this before, but anyway, this one, even though the name is different, basically is similar to the probiotic. The symbiotic is defined as a mixture of probiotic and prebiotic. So basically, it's a probiotic plus the prebiotic that beneficially affect the host by improving the survival and activity of beneficial microorganisms in the gut. Okay, symbiotic were developed to overcome possible survival difficulties for the probiotic. And then, so basically, the probiotic strain candidates will be the same, like the lactobacilli, bifidobacteria, streptomyces, like that. But you add with the major prebiotic, such as the oligosaccharides or inulin. So basically, this one, the function is almost similar to the probiotic just now, how it works, but the supplementation is actually a two thing. One is probiotic, one is prebiotics. Okay, now, just now we talk about the interaction between when the disease occur. If there is a three ways of interactions, how to control it. First is the pathogens outside the culture system. Then the pathogens is inside the culture system. So we need to administer the prevention method. So the third interaction is when the disease occurs. These things is when the pathogens already cause the infections to the pathogens, to the fish. So basically, we need, for this one, we need to do treatment of the disease. So right now, when the disease occurred, we cannot use vaccine already. Vaccine is meant for healthy fish. So if the pathogens already resides in the fish and cause the damage to the fish, so we need to treat it. So previously, people use drugs, but due to the many things, because currently in Malaysia also we already been prohibited the usage of the drugs for the food fish. So we need to find the alternatives, actually. So that's why it comes to the biocontrols. We have the biocontrols for the preventions, like the probiotic, the immunostimulants, but it's very difficult to find the biocontrol for the treatment. So here I list the biocontrol, some of the biocontrol for the disease treatment, and also some of possible biocontrol that we can explore as the to treat the disease in the aquaculture. Here goes. Okay, so for the first biocontrol is the materials, the way of action similar to the antimicrobe, meaning that the functions or the how it acts is similar to the antibiotics. So what are these things? There are two, actually, based on the previous study. First, phytobiotic. We can use the natural plant extract as a substitute to the antibiotics. The other terms for this is phytobiotics. Many plants are known to have anti-stress, growth promoter, appetizing, tonic, immunostimulations, antimicrobials, antibacterial, and aphrodisiac properties. But we focus on the antimicrobials and the antibacterials. So these properties, however, are linked to the presence of bioactive compound. So this bioactive compound which that cause the antibacterial or antiviral or antiparasite or antifungal. Such as the alkaloids, flavonoids, pigments, phenolics, terpenoids, steroids, and other essential oil. Some of the plant reported to have antimicrobial compound that able to suppress the growth of aquaculture bacterial pathogens, of course. So previous study, of course, the extract from the beetle is sirih. Stormbaker, I'm not sure what is this plant. I'm still looking for it. And then the clove is the cengkeh. So basically this one, the extract from these materials can be used to inhibit the bacterial pathogens. I believe there's a lot of information on the other candidates of the herbs or any plant, or even the seaweed from the marine environments. We can use that as well. And we already test it against the aquaculture fungal pathogens. Then it can, in the lab, they can suppress the growth of the fungi. So this is what first biocontrol strategies. Second is the postbiotic. So the postbiotics, another way to say this one is actually also the extract. Okay, the extract from the bacteria, live bacteria. Postbiotic refer to soluble factors, products or metabolic byproducts secreted by the live bacteria, for example, from the probiotics, or release after bacterial lysis, such as enzyme, peptides, tectonic acid, peptidoglycans, these etc. things, such as protein and organic acids. So these properties from the bacterial lysis or release from the live bacteria, they have the antimicrobial properties. Currently, we have tested it onto the bacterial pathogens and also fungal pathogens. Then they have the antifungal and antibacterial properties. So the examples of the postbiotic is bacteriocin. Bacteriocin is type of the antibacterial peptides released by the bacteria. Usually we use the lactic acid bacteria. There are some reports that showing that the bacteriocin, they also able to act as the antiviral, antifungal, but very limited information on the anti-parasite, as an anti-parasitic. So basically how it works for these things, when I talk, it's similar to antibiotics. It will cause mortality to the pathogens. So basically the pathogens will be dead lah when they mix with the postbiotic. They cannot survive. And then there's another information for the phytobiotics, whereby they have abilities to inhibit the quorum sensing. Quorum sensing is the method of communication between two bacteria or a group of bacteria. So basically, when the bacteria reach certain concentrations, they will communicate and release the toxin factor or the virulent factor together. By inhibiting the way of communications between the two bacteria, even though they reach certain concentration that able for them to cause the infection to the fish, but due to the inhibitions of the quorum sensing, they are unable to infect the fish. So this is the other information about the phytobiotics. Okay, so for the biocontrol, next biocontrols that we can use as an alternative for the treatment is through the predator-prey relationship. Just now is inhibitions, they able to kill the bacteria or the fungus or the pathogen directly. Okay. Now the other biocontrol is through the predator-prey relationships. What are these things? Here I show you guys 5 examples. 2 of them is being proposed in aquaculture. The rest is still under concept. Okay. First, for the predator-prey relationships, in order to find them, we need to know the natural enemy of the pathogens. What are the things that are able to eat or kill these pathogens, meaning the living microorganisms lah. Because these pathogens is very small, so it needs something that also small to counter them. So first, of course, bacteriophage. For the bacteria, the interest in phage therapy has been revived in recent years following the rampant occurrence of the antibiotic resistant bacteria. So bacteriophage is actually as a virus that able to infect bacteria. So by infecting the bacteria, they cause lysis to the bacteria and the bacteria will die. So in aquaculture, bacteriophage has been used to control pathogenic bacteria. Due to their ability to kill target pathogens, they become a tools for treatment of infectious bacterial disease. Here, the bacteriophage is only specific for the bacteria only, and even species specific. So for the other pathogens, we cannot use the bacteriophage to attack them. For examples, the VIROPHAGE is a type of virus that prey on Vibrio species. So basically, you guys know that in aquaculture, Vibrio is one of the famous pathogens in aquaculture. Next, cleaner shrimp. So this one is something that able to against the parasitic infections. Cleaner shrimp preys on benthic stages, suggesting they have the potential to reduce parasite reinfection pressures. The cleaner shrimp have never been used as biocontrol in commercial aquaculture, but they offer an advantage as alternative to reduce parasitic infestations. For example, Lismata, this is the name, genus, species name of the cleaner shrimp here. Some paper like from the Millis and Hudson, they have tested that these cleaner shrimp consume the eggs and larvae of the harmful monogenous parasite. This monogenous parasite is actually one of the parasites in aquaculture. So basically, this is one of the candidates also. The rest of the candidates that I will show later, it's only still under concept, but people already study. These things exist in the environment actually, but never been tested in aquaculture. Here goes. First, praying bacteria. Just now we go with the virus that able to eat bacteria. This is the bacteria that eat on bacteria, but I won't say this one is cannibalistic because they are different species. Different species of bacteria will infect the not infect it on other bacteria. We call it praying bacteria. The praying bacteria or predatory bacteria act by kill and consume their prey. But here, if you guys look into these informations, it's not that the praying bacteria is bigger than the pathogen or target bacteria. Is actually smaller than the pathogens. For examples, this is the species, genus, basically genus Bidella vibrio. This is the name of the species that able to prey on many types of bacteria in the environment. So basically, they have the potential to become candidates. Besides the bacteriophage and also the cleaner shrimp to replace the antibiotics. Okay, next, virophage. Okay, as the name stated, this one is the virus that eat on virus. Well, virophage is a small double-stranded DNA virus that parasites of giant DNA viruses. Basically, this one is, I think, is just recently published. The thing exists, but of course never been reported related to the aquaculture or agriculture. Basically, it's quite new. This one is quite new. The name does not signify a virus that infect other virus. It means the virus eater, meaning that the virus eat other virus. So this one, by this concept, we can, if possible, get the virophage for the pathogenic virus in aquaculture, then that one can be a candidate to control the viral infections in aquaculture. So for the examples, if you want to check on the concept of the virus infecting or eating other virus, you can look into the Sputnik virophage. This is, I think, this published by this Mogari et al. 2019. Lastly, okay, fungal parasites. Okay, the fungal parasites meaning that predations by the zooplankton on the free swimming zoospores. Usually when we talk about zoospores is the infective stage of the fungus. This is where the if they want to infect the larvae, they need the zoospore. They use the zoospore. It's not from the mycelia. So this predation has been suggested as a possible method for biocontrol of pathogenic fungus, but not in aquaculture yet lah. Haven't. So based on the study from this here, okay, I hope I pronounced the names right. Daphnia species can reduce the density of the parasitics, okay, the parasites. This is with fungal. Batrachocriterium. This one is a parasitic fungal to the frog actually. So the Daphnia, they can reduce the density. So after they reduce the density, they maybe have the possibility to reduce the infections from the fungus. So maybe this one we can use this concept to apply it in aquaculture. So as for conclusion, thank you so much for hearing me just now. So basically we need to explore alternative to the current control strategies. What I mean is current control strategies is the things that we have now, either using the chemicals, of course, no antibiotics. Okay, and then the informations maybe hope it provide the insight to the possibility of using the biological control to suppress the infections, especially the treatment, because we don't have any method of treatment currently in aquaculture. Okay, besides that, I would like to take this opportunity to give some information to everyone. If you have any questions regarding my topic right now, if later, you may contact me through the email, or you may contact me through my mobile phone. Basically, this is the only two ways two way of communications now. And in fact, that our institute is a postgraduate institute. So if any of you want to pursue study in a master degree or PhD degree in the aquaculture, marine science, fisheries science, and marine biotechnology, please give me a call lah. So you can contact me through the email. Okay, I think that's all from me. Thank you so much for listening. I pass this back to the moderator.
Thank you very much, Dr. Tamrin. It's very interesting presentation. So you explained about the biocontrol for environmentally friendly. So maybe we have some question here from direct message to me and also in chat room box. So maybe we will bring it up in discussion session. So now we are going through to the speakers too. We proceed to the second speaker, Dr. Rofi Grandiosa from UNPAD. Mr. Rofi Grandiosa, hello.
Yes, thank you. Hi, everyone.
Okay, before that, maybe I will read the CV of Mr. Rofi Grandiosa. He is a lecturer of Faculty of Fisheries and Marine Science, Universitas Pajajaran. And he also graduated from Auckland University of Technology in Applied Environmental Science. And also his master degree is Faculty of Bioscience and Engineering in Jan University in Aquaculture. And his expertise is aquaculture, immunology, and pathology. So without further ado, Mr. Rofi, are you ready?
Yes.
Yes, okay, screen is yours.
Wait a minute. Yes. Okay, good morning, everyone. My name is Rofi Grandiosa, and I am a lecturer in the Faculty of Fisheries and Marine Science. So today my talk will be the Virtual International Aquaculture Talk, between FAK UNPAD and Borneo Marine Research Institute, UMS. My topic is status of fish disease and health management practices in rural freshwater aquaculture in West Java, Indonesia. So this topic is based on my experience and some of my past research. And let us go on to the next slide. The order of the presentation today will be introduction, aquaculture in West Java, current issues of fish disease in West Java, recommendation and conclusion. Okay, West Java, where is it? It's highlighted in the red here. See here. So West Java itself is where our university is located. West Java itself has 18 districts and one of the densely populated province in Indonesia. Currently is around 49.9 million people residing in this specific region. And UNPAD, place I work for, is one of the largest national university in Indonesia, located in West Java. Here is the logo of UNPAD. Here is the logo of coat of emblem of our province. Talking about West Java aquaculture, we start first about aquaculture in Indonesia. According to the recent, or not so recent, FAO 2016, Indonesia is one of the top 10 aquaculture producers in the world. In 2014, Indonesia is number 2 with a total aquaculture production of 14,330,000. And from that amount, 2.8 million comes from inland aquaculture, which may consist of freshwater aquaculture and brackish water. So West Java, there's a lot of things going on, especially in freshwater fish culture. Aquaculture in West Java, according to the recent data in 2018, there's a annual production of 1.1 million tons of fish. And how many people are involved in this aquaculture practice? So around 600,000 persons. The main species, especially for freshwater, is we can say there's a common carp, Cyprinus carpio, tilapia, and then tawes, Pontius javanicus, Nilam, Osteosus hasselti, African catfish, Carius garipenus, Pangasius, the Pangasius hippophthalmus, and the gurami, giant gurami, which is the Osporenmus gurami. Here we have some pictures, common carp and tilapia, so on. Yeah, since we are talking about the issue of fish disease, fish health, the current issues of fish disease in West Java, of course, there are a variety of infectious agents which are present in aquaculture activity. So farmers in rural areas usually find culprits caused by bacterial infection mainly. Other than bacterial infection, there's also, of course, virus and parasites and fungal parasites. But usually, the main disease causing mass mortality is opportunistic pathogens such as Aeromonas hydrophila, Pseudomonas, Ateromonas, Streptococcus, and 4 catfish strains or Pangasius edwardsiana. In general, the rural aquaculture community understand the concept of fish well-being. However, the knowledge of immunology, medication, or disease prevention may be a little bit lacking. Yes. Example of catfish with the disease, bacterial disease. So current issues in freshwater aquaculture, which I observe for example, past 10 years, is a pathogenic disease. So let's say on common carp, in the past there was a very large outbreak caused by KHV, koi herpes virus. This caused very high mass mortality, and also other bacterial disease accompanying this virus, such as Aeromonas. And then for tilapia, there's also this kind, Streptococcus, Aeromonas, and recently the issue of tilapia lake virus, for example. In catfish, there's bacterial disease such as Aeromonas cerevisiae, gurame. Gurame in West Java is very highly cultured because it's a very important species for certain occasion. Gurame is consumed in Indonesia. However, during culture, for example, in the juvenile stage, it is very highly affected by fungal disease such as saprolegnia, causing mortality, high mortality, and also reaching larger size. There's also Mycobacterium, and then again, Aeromonas. This is some kind of opportunistic pathogen. Other common pathogenic agents such as ectoparasite and endoparasites are also present in the ecosystem. Yeah, current issues of fish disease in West Java. Actually, in West Java there is a reservoir. There are 4 reservoirs currently in running along the Citarum. There is Cirata, Jatiluhur, Saguling. Sometimes there is mass mortality in occurring. Maybe not directly caused by fish disease, maybe caused by environmental problem. There's also a new reservoir, very large, in the Sumedang district. There is a new reservoir. And of course, the farmers who open cage aquaculture in this area are, as Muhammad Tamrin explained, diseases are present in the lake. If we culture in the cage culture, of course, fish are directly affected by pathogens present in the open waters, for example. Of course, other than open water aquaculture, we also have culture in land-based, in earthen ponds, in tarpaulin ponds, and recirculating systems. So usually the rural community, this is myself, I did a service in Pangandaran. Yeah, they really need information how to keep their fish healthy, for example, and they're really eager to know how to maintain their water quality, their fish health, and all aspects of aquaculture. Mainly fish disease, because when fish starts dying, usually the farmers sometimes give up or desperately take actions, for example. Yeah. So regarding the fish disease in West Java, this is based on data from the fisheries local government in West Java. The use of medication is quite high in the yearly use. In 1 year, they could use around 6,000 tons of medication. There's a five aquaculture system identified: still water, running water, cage culture, rice field, and floating cages. All the system were recorded to use medication. I don't know what kind of specific medication, but huge amount. Yeah, in still water 6,000, in running water 1,400, and in rice field 5,400. And also they use pesticide and to remove, for example, parasites. Yeah, parasite possibly, and disinfectants in the beginning of the culture system, and also hormones. So these are the data from. Thank you for the government, for the fisheries local government. I have my friend in one of the participants, Mr. Dede, yeah, from Java, West Java. And then current issues of next, probably the common issues was probably the usage of chemical medication, such as antibiotic. We could find in the marketplace, there's a lot of medication, which is according to, yeah, it's a common sense nowadays. I guess Dr. Tamrin agree. It's very dangerous to use antibiotic, but it is available in the marketplace for example, Tokopedia in Indonesia. Yeah. You can just buy it from online and receive it and do some things without supervision. But in general, farmers who are supervised by the government, they usually visited by the government officials and the government official, and even the university staff. We contribute to community service to give knowledge about the danger of antibiotic and everything. Yeah. So the general rural aquaculture community understand danger of antibiotic, and of course all stakeholders will continue to disseminate strategies for a better environment, because we know that sustainable aquaculture is the key for the future. We need to have constant production for healthy products. So, of course, applying standardized fish health management prevention practice is needed here. And then current issues of fish disease, again, how does rural community understand health management and fish disease? It's mentioned also by Dr. Tamrin, there's traditional herbs in research, but actually it's a local knowledge because farmers in the rural areas also know the advantage of herbs such as papaya leaves, and then the piper beetle leaves, as you mentioned, sirih in Indonesia, and also all kinds of leaves. My students in the past also studied that, dry the extract. This traditional herb, of course, can cure fish, phytobiotics. Number two, probably information, not probably, this is activity is going on, local government directly disseminated. There's officers in the field who are, the salary is paid by the ministry, if I'm correct. Yeah. They are located in various parts of rural areas. These people, these officers will inform farmers how to culture in good way, how to keep everything going on as business, as a profitable business for aquaculture. And then the third point, probably monitoring of fish health through quarantine gates. For example, one of my students is culturing koi fish. Yeah? His market is domestic market. He transport to Manado. He gets information about disease from quarantine gates. So there's many quarantine gates throughout Indonesia. So if you want to deliver a product to another place, usually they have to put it through quarantine. And then also certification of aquaculture. Yeah. In international is good aquaculture practices, but in local version it's CBIB, cara budidaya ikan yang baik. This certification will enhance or increase the reputation of the farmer, because suppose the farmer will have better culture knowledge and healthier fish probably. And then there are also the role of private company. Some of my students also work as salesperson for feed producers, fish medication salesperson. They also have the role to inform disease and how to cure fish in the field, for example. And the last one, user-oriented information. So the rural community are, of course, nowadays in 21st century, they're really connected to the internet. They could search Google and watch other people doing, other farmers curing fish through YouTube, and they could make a group in Facebook, yeah, and also they could, nowadays they are really eager to actively participate in meetings such as this one, webinars. Yeah. So this is introduction. So the rural farmers are really eager to know how to culture, because sometimes they're, yeah, example, yeah. And maybe to the next slide, the rural community needs more understanding, since culture of fish may be more complex than it seems to be. I think, yeah, for us, maybe it's easy to understand immunology or how to disease management and everything. Yeah. This takes a long time to disseminate. Yeah. But the rural community is the key for success for aquaculture in our area, since fish consumption is very high and ongoing population in West Java. Of course, fish produced in West Java also is marketed to other regions, to Sumatra, to Kalimantan, and to other parts, because even the common carp from West Java is really liked by the Manado. It's very far, but they like fish from West Java, the common carp, for example. Now, how does our campus and other campus participate? So we have many universities in fisheries, aquaculture. There's UNPAD, there's IPB, ITB. How do we collaborate? Yeah, usually we initiate community service for literacy and holding seminar, actively participate in seminar groups, research group. There's a lot of things going on. And each researchers also doing research to develop new things such as vaccines, herbal medicine, probiotics. Actually, vaccines, Dr. Tamrin mentioned vaccine. We also, my colleague in Bogor also developed a vaccine to prevent Romanes. And then there are also people doing, developing disease-resistant strains of carp. So they breed intensively, breeding common carp and make it mantap. Yeah, mantap means what you call it? Majalaya tahan penyakit, which is one of the strains of common carp. Majalaya tahan penyakit, and then they make it the term mantap fish. It is disease-resistant fish. And then we need, of course, all the stakeholders should be actively coordinating with government officers in quarantine units or local fish centers. So other than quarantine units, throughout West Java, there's also many fish centers who are, for example, one center only focus in tilapia and another center focus in catfish, for example, like that. So many fish center with specialized species developed. And recommendation from myself through the knowledge of fish disease I acquired for the long time. Yeah, I think recommendation is to disseminate knowledge from researchers. Sometimes the knowledge is just kept in the university. This knowledge should be further disseminated with simple words and simple techniques, so all the rural community could understand better. Improving fish seed quality is done through breeding programs. I think this is an ongoing program done by all stakeholders. Improving good aquaculture practices program with the local version of CBIB, as I mentioned in the previous slide. And probably knowledge upgrade of fish health for responsible aquaculture community service staff. Yeah. Sometimes they're just newly graduated probably, and the knowledge need to be upgraded, constantly upgraded. And then one of the things that is important is transboundary fish disease. Yeah. So we need to reduce transboundary fish disease through strict quarantine protocol, such as tilapia virus. This disease was I think it's one of the transboundary fish virus disease. Yeah, for example. And KHV also, KHV virus is transboundary. So a lot of things, achievements in the field are present. I think fish are more healthier, however, yeah, needs to be more improved. Conclusion, fish health remains an important issue, not just in West Java, in all Indonesia. Since fish is an important source of protein other than from animal husbandry. Fish health remains an important issue. And then the knowledge of rural communities still needs continuous improvement. Yes. And then the third point, better knowledge of fish disease should be continuously discussed, such as in this forum, because in the Southeast Asian aquaculture community, we prioritize since we share similar culture of aquaculture preference and practices. Okay, thank you. If you want to catch to me, you can see my LinkedIn, Google Scholar, ResearchGate, Rofi Grandiosa. My email is written there, rofi.grandiosa@unpad.ac. Thank you for your attention. I will give it back to the moderator, Dr. Fitri. Thank you.
Thank you very much, Dr. Rafi, for the presentation. So you have presented about the special issues about fish disease in our province in West Java, how it happens, and also how to overcome these problems. Yeah, we know that there is no in West Java only, but in maybe in several rural communities, we have also this problem. So based on the expertise and background of two speakers, in this section, this discussion session, we have several questions from audience, both in a chat room box and also private message to us. So, okay.
Yep.
Okay, there are some questions here. Wait a moment. Okay, there is a question from, okay, this is a question from audience to Dr. Tamrin. Okay, regarding about the vaccines, what kind of product have been developed and what fish and how to prevent and what kind of disease, especially in Malaysia. How does Malaysia prevent a transboundary disease? Maybe we ask to Dr. Tamrin, and you can see in this screen, Dr. Tamrin.
Okay, thank you so much, Dr. Fifi. Where's the question just now?
Oh, sorry.
Okay.
Okay. Okay, please.
I think the first question is about the vaccines, right?
Yes, the vaccine.
The vaccines. So currently, in here in Malaysia, we don't have any commercial vaccines yet. Basically, so basically, majority in aquaculture, usually the vaccine is come from already being developed in other countries such as Korea, China, or other Europe, and also the Europes. So basically, for the common vaccines that have been produced for the time being, to my knowledge, is focusing on the virus and also the bacteria. So this is the only vaccine that is available in the commercially. So I think that one is if I want to answer the first questions just now. Right. I don't remember the follow-up questions. Do you mind to show it again so that is
yes, sure.
Sorry everyone, I cannot remember the
wait a moment.
question just now.
Okay, so There's some questions,
Okay.
especially for you, regarding about the vaccine, and also
Oh, I
if there is a
see.
vaccine, what kind of product have been developed, and if not, it's okay, and what kind of fish. But how Malaysia, especially Borneo Marine Research Institute, to prevent of disease in Malaysia?
So, okay, maybe I have to refresh back about the questions on the for in our institute. This one is quite new about the study on the vaccine. Currently, we have just very limited information on the trial on the vaccines for to against the very specific disease, which is fibrosis. This is one of, I think, very devastating disease in here in marine industry here. Okay, so that's the apa information that we have so far. Currently, we haven't developed any commercial vaccines for the to against this the pathogens or for the against this fibrosis. Okay.
And also a
And
second question, maybe. Okay. So there is a question from Fahri. Wait a moment.
I think about the biocontrol, I cannot
Yes.
remember.
Oh, yeah. There's about the biocontrol. Biocontrol for treatment in fish.
Okay, so about the biocontrol to the treatment, basically this I think is almost similar to the like the previously if you use the antibiotics, they are withdrawal period that we have to wait before we able to sell the fish. But obviously now we cannot use that. So if we want to use any biocontrol methods such as the extract from the plants, thing, usually for any kinds of the biocontrols, of course we cannot give the feed the extract to the fish, and then next day we sell the fish to the consumer. So of course there's a withdrawal period. Currently the safest death period usually is 1 month. So basically that's a, I believe with further study on the withdrawal period for each of the species of the fish, maybe we can get the meaning correct or accurate withdrawal times, because if for the natural products, usually they can be digested or they can be degraded easily compared to the synthetic antibiotics. So I hope that one answered the questions.
Yes, yes. Maybe generally, the practical use of a biocontrol, or maybe some antibiotic or something like that, maybe similar between Indonesia and Malaysia, I think. And there is one question from participant, from Mr. Untung Kurnia Agung. He is from Fisheries and Marine Science faculty. He is a lecturer. He has long question. Maybe to Mr. Untung, could you like please to open your video and you ask by yourself or something?
Okay, I think I can see the questions in the chat
Okay.
box.
Okay.
Okay, so
Yep.
no, so this one I can read. So never mind. So the first question is about the because of many kinds of pathogens that we can control to infect the fish, that may be there against the fish at the same time. Is there any multi-pathogen prevention system using vaccine, probiotic, others that effectively apply in aquaculture? So basically, I think that the currently not the best, I would say, is better prevention method to against the disease currently is using the probiotic. So far, the probiotic not only they have the ability to outcompete the growth of the pathogens, meaning that they reduce or control the volumes, the concentration of the pathogens. They also can facilitate in terms of improving the growth performance of the fish. That one I think has been been mentioned in many journals. But that one, so far for the probiotics, of course, the bacteria is the main target pathogens they want to control. So far, I have read some informations that the probiotic can be used against the pathogenic fungus and also the virus. These things have been reported in the literature review. But about the parasites, it's very difficult currently. I haven't found anything. So basically, about the materials that we can use to target all of this, it's very it's like currently maybe they are, but we haven't found it yet. So as a researcher, it's our job to keep on researching and searching for these things. Okay, I hope that one answered the first questions. And about the second questions about the phytobiotic and quorum sensing. Okay, whether the actions of certain phytobiotic in interfering okay, quorum sensing is specific to particular bacteria species, or is it applicable to many species?
So many different species
Okay.
of pathogenic bacteria.
Based on the questions, when we talk about the quorum sensing, I think this one is more on the, because in the plants or from the plant extract, they have many compounds. So these compounds, some of them maybe have the ability to inhibit the signals from the bacteria. Of course, the bacterial species have different signals, meaning that when they want to communicate, they will release certain signals, and these signals will be recognized by their own kinds. So I believe that, of course, if we want to use specific extract, meaning specific compound, maybe this thing only able to control certain species. But if we use the plant extract like the crude extract, because there are many metabolic compound inside that, so different metabolic compound from the plant extract may be able to inhibit different species of the bacteria. Okay, so I think yes, it is applicable to many different species of pathogenic bacteria. Thank you.
Okay,
Thank you for the questions.
Thank you, Dr. Tamrin. Maybe the answer will help to participants to understand about the knowledge of this biocontrol. So there's also several questions to Dr. Rolfi from some participants. Okay, the first question maybe, okay, I will read it from Dr. Ching Fui Fui. Dr. Rafi, are you there? Okay. Is the use of SPF broadstock being practiced in Indonesia or not? And then the second question from Aisa, this is student also. From all fish disease cases in West Java, what kind of treatment have been applied effectively and how to prevent these dishes?
Okay, I would like to answer the question from Dr. Hui Fui first. Okay, SPF broodstock, probably your question is related to the Faname species. Am I correct? The Faname? Yes, Faname, I think, developed by the company in Indonesia, in Universitas Pajajaran. Actually, I'm not, what you call it, the expert in this field. There is Dr. Asep Anam. He worked in shrimp improvement system, who bred Faname in Hawaii. Yeah, there's one of the specialists in working in UNPAD. So yeah, the SPF broodstock there are in the companies, private companies of Faname. What in my presentation, probably the disease-resistant fish, yeah, common carp, it is applied, but it's not pathogen-free. It's a disease-resistant for carp. But for Faname, Panaeus Faname, it is existing, and it's. However, yeah, the Faname industry is currently, there's a lot of things going on. The disease of what you call it, acute disease is happening also in Indonesia. Yeah, APHND. Yeah, is also happening, and yeah, a lot of things, a lot of topics. Maybe for a different time, Dr. Fui. The other question is from Aisha. What kind of effective strategy, for example, in West Java? I think the most active the strategy is keeping the fish healthy, of course, with a better standard operating procedure. For example, the knowledge of preventing is better than curing. Yeah, I think having better quality fish, and then when there's disease, of course, we need to have quarantine protocols in the aquaculture side. So what I observe from my experience in New Zealand, in Belgium, they have a good standard protocol to prevent disease, yeah, and keeping fish healthy in general. I think that's my question. Thank you, Dr. Fitri.
Okay, thank you, Dr. Rafi. Maybe this answer will help to give a good answer for Dr. Singh Hui Hui. It's okay for this answer. Okay, thank you very much. Maybe there's no question here, or there's a question, one question. Maybe it's okay. Or if you want to ask directly, you can raise your hand and activate your microphone and also the camera. Okay, maybe not. So let me wrap up this discussion session. So this time we have a theme of fish and disease management practice, and based on the Dr. Tamrin materials, there's a biocontrol who can save and also environmentally friendly to prevent and also to reduce the synthetic chemical from such as a vaccine and etc. And also Dr. Rofi also show us the common issues in the West Java, especially disease in rural community of aquaculture, and also the usage of chemical medication such as antibiotics in rural communities in Indonesia. So maybe this discussion will help us to understand about the fish disease and health management in aquaculture practice and how to develop the best aquaculture practices. Thank you very much to Dr. Tamrin. Thank you also to Dr. Rafi for your excellent materials. And ladies and gentlemen, we finally come to the end of the part of this session. So, first, don't forget to fill in the link of form attendance of today's talk. And I'd like to thank you so much to the speaker for informative and interesting presentation. And also, please give applause to the speaker and for you all. See you next time and have a nice day. Thank you.
Nggak ada foto, Neng Fitri? Foto bersama?
Next session.
Oh iya.
After this.
Ladies and gentlemen, we are very honored to have with us here this afternoon. Before we close the international virtual talk, would you please to turn your camera on to take the virtual photo session? Have everyone turned your camera? Okay, let's start taking picture. I will start from 1, 2, 3. Okay, once again. 1, 2, 3. The last one. 1, 2, 3. Okay. Thank you very much, everyone. Ladies and gentlemen, by the end of this photo session, it means that our virtual talk today is closing to the edge. Before we close it, I would like to inform you that in the chat room, there is a link for your attendance today. Please fill it up because we will send the certificate and the materials of today's event to your email based on this data. In addition, I would like to inform you about the International Aquaculture Talk Series 2. The schedule will be on Thursday, February 4th, 2021, with the theme of application of genetics in freshwater fish. So don't miss it. Thank you very much for your attendance. I am Adinda. See you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you, Bu Yuli. Nuhun.
Wilujengnya, Vi.
Thank you everyone.
Ya,
Thank you for
Thank you
having
semua,
me.
tahniah.
Everyone.
Thank you Dr. Siti, Dr. Kui.
Tahniah semua, hebat-hebat.
Ya.
Mantap,
Thank you,
semua
Dokter.
hebat.
Jumpa tanggal 4 ya kita.
Ya.
Yeah,
Hebat
next week.
semua. Hebat
Ya.
semua.
Iya. See you,
Oke,
Dr. Siti
selamat.
and Dr. Muhammad Tamrin. Terima kasih semuanya.
Terima kasih
Dr.
semua.
Siti,
Ya.
Dr. Tamrin.
Sama-sama. Thank you,
Ya,
Dokter.
Dr. Iskandar semua. Tahniah semua ya, tahniah.
Bye-bye.
Bye.
Okay, bye.
Alhamdulillah. Nuhun ya. Terus hebat.